In this video, we're going to be focusing on the testing part of the scientific method, the fourth step, where you actually attempt to disprove your hypothesis by doing an experiment or a correlation of study. Whichever one you did, there are certain criteria that I'm looking for when you write up your lab report on what you've done. And this is what we're going to be talking about now. Now, this includes both the materials and procedures section of your lab report. Now, for the materials section, I'm going to be looking to see that you've listed every single thing that you've used, including the quantities, because I need to be able to replicate exactly what you did, and that means that I need to know exactly what you used. So if you use some sort of apparatus, if you use some sort of device, it's important for you to include a description of that device and instructions on how to use it and the configuration of your lab setup. It's also important to you tell me all the parts, all the tools and utensils that were used. And if you did this on people, it's important to describe the population that you used. Let's say you did a psychological study, for example. I want you to know the number of people in each sample, how the sample was acquired, the demographics, which includes gender, the ethnicity, the country of origin, the language, the socioeconomic status, the age of the group. I need to know the composition of the people who were involved in the study. Now, when you come to your procedure section, you also have to write these procedures in a full paragraph format or in bullet points that address exactly what you did in a step-by-step format. The best way to do this well is that when you're doing things in the experiment, to have someone who's there writing every single thing that happened and every single thing that you used and did. That way, when you actually write your lab report, it should be easy to do it because everything you picked up, you wrote in the materials section. Everything you did, you wrote in the procedures. And then when you do that, it'll be easy for you to do this section of the experiment. So it's very good to have a data manager that's taking care of that when it comes down to the experiment. Now, when you're writing your procedures, I'm looking for these things to be included on your procedures, okay? The 5 basic parts of the experiment, plus a few more things. I'm going to need to see that you identify your independent variable and explain how are you going to be manipulating it. That is the experimental process. Basically, what are you doing to the independent variable? How are you changing it? Okay? So in the example that we've been talking about, she's going to be changing the amount of acid or the concentration of the acidity in the samples. So she's going to have to describe how she makes each one of those samples. Just like in the materials section, she would have to list every single chemical that she used, every single vial, the plants, the pots, everything that she did would have to be included. In terms of the dependent variable, you kind of have to explain what's your dependent variable and how you're going about measuring it. How are you collecting this data? In the case of the experiment that we're looking at, I'm looking for something like I'm going to get a ruler, and I have to describe this ruler, of course, and then I'm going to say how exactly I'm going to be using this ruler to measure. In their case, I think she's using the vertical height of the plants as an example, or the extension from the root to the tip of the plant. And so you also have to have evidence of the control and experimental group, because remember, you have to have a group that's being treated and a group that does not directly receive treatment, because any change that happened in that group has to be compared to changes happening in the experimental group. The experimental group has to change more than the control group for you to be able to say for certain that the experimental procedure actually made a difference. So it's important to have that within your experiment. I'm also going to be looking for efforts that were made to maintain things constant other than the independent variable. Remember, only one thing can change at a time unless you're doing a multifactorial design. And that means that you're going to have variables maintained constant, such as, for example, we talked about this when we did the background, the pot that you use, the amount of water, the exposure to sunlight, the type of plant, and all the other things. And I want to see in your procedures that you specifically mention things that were held constant on purpose. You know, those control processes. I'm also looking for safety and ethical concerns. Is there anything about this experiment that could cause ethical problems, or anything about this experiment that could be dangerous in some sort of way that I have to know about, or that people involved in the experiment should be aware of? Also looking for steps that you took to maximize reliability or validity, accuracy, precision. Some of these things could include: how did you take samples, especially if you work with people? How did you sort people into different groups, experimental or control groups? Did you have repetition? Did you take averages instead of just measuring things once? Did you have blinds or placebos if you had to do that? Did you make efforts to use the best tools and methods available? Did you compare your results to those of other people to make sure you could, the validity and generalization of your experiment was good? All of these things should be included. You should also include how you intend to analyze your data after you collect it. You should say, what are you going to do with the data after you finish collecting it? All of these things should be included within your procedure section, and that's how you write a good one. So let's go look at the sample that we have along with the rubric to see if we have what we did. You should spend some time, pause the thing to read the lab report and before we talk about it so you can see what's going on. So looking at the rubric, I'm looking for specific descriptions of all materials used. So now upon reading her procedures, I noticed that she mentioned that she had to boil water. But I don't see any indication in her materials of what she's going to be using to actually boil the water. She mentions a small pan, and I'm guessing that she's going to put the cabbage in a small pan and then put it on a stove, but she never mentioned to me how exactly she's doing that. So right there, she lost those materials points for not putting all the materials used. Now, list drawing of all laboratory apparatus used in the investigation. You know, I don't see that here either. I do not see any indication of the apparatus that she used. And so she would have lost that point as well for not including some sort of description or picture that showed the apparatus that she had to set up, to do what she had to do. She didn't do a population study, so the M3 does not apply. And she didn't list all the quantities used. That she do, so she should get that material point. So see, it's not easy to write a material section. It is, though, when you're doing the experiment, you write everything down. Then you don't find any problems. Everything you've used, anything you touched, you write in the material section, you'll be fine. Now, when it comes to the procedures, did she write in bullet form or complete paragraph format? Yes, she did. So she should have got the P1. Did she mention all the steps and details of her protocol? I think she did a pretty good job. I have a good idea of exactly what she did. I don't think it was perfect, but I think it was really good. So I would have given her this point. Now, she indicated how the dependent variable is going to be measured. Yes, she says measure and record a plant's height daily in centimeters. Now, you could argue that she never really explained how you measure a plant's height. I guess she thought it was self-explanatory. A good experimental design should include exactly clear definitions of how you're going to be measuring. Something like, for example, measure the distance from the bottom of the stem to the topmost part of it, in centimeters. But, this is being picky, so I think I would have given her the point in that case. But remember, when you're designing your experiment, that you should try to write as much detail as possible. Indicates how the independent variable is manipulated. So let's look at that. Does she talk about different groups and how she changed those groups? Yes, she describes how she created the different acidity levels, and she also described how she would use the red cabbage indicator and how to even make the red cabbage indicator. So that's actually very cool. She actually has all of that. Does it indicate a presence of control versus experimental groups? Yes, she talks about having a group where she doesn't put any acid. It's just a control. So that's good. Did she talk about standardizing variables or things that she should maintain constant? Not directly. She doesn't talk about directly that she's going to use the same bottle, the same... she doesn't talk directly about it, but there are some indications of things that were going to be kept constant. But it's better if on your materials you describe clearly all the things that were made efforts to be held constant. I think I would have taken this point off because she didn't clearly stipulated that. Includes safety or ethical protocols. She's playing with acids. She's playing with boiled water and all these things. And so she should be careful about that. And she should also use eye protection when using these devices. I see some things there, but she didn't talk about, boiling the water carefully and things like that. And I don't think it's a complete safety protocol. So maybe I would have taken that point off as well. Now, includes steps taken to maximize validity, reliability, or precision. Now, that includes repetition, tools, things like that. Now, She never mentioned to me that she was going to use the most accurate type of ruler that she could, but she did say she was going to do it in centimeters instead of inches, and that's good because centimeters are more precise than inches. But she could even have done it in millimeters instead of centimeters. So I think that's one thing she could have done better in terms of precision. But in terms of pH level, for example, she didn't probably use the most precise tool she could. She used cabbage pH indicator instead of using electronic pH indicator. So, but I mean, she did everything she could. She's a student, you know, so you have to take that in consideration and be nice. I don't think I would take the point off for that. Also, she did mention in her experiment that she was going to have several repeated studies, so she would measure every single plant. She was going to choose 3 for each kind. So that means that she's actually repeating the experiment, so that's good. She also does not include a description of how she's going to be analyzing the data within her procedures. And so she will lose the last point, which is that she's supposed to include that on her procedures. So that's how you write a good procedures and materials section. And now I hope you understand that and move on from here to talk about analysis and results.